Thank you very much. I would like to thank Anne and the team, organizing team, for inviting me to this Education Day program and for giving me the opportunity to talk about extracellular vesicles and particles. I would like to start with the nomenclature. According to the MICEF 2023 guidelines, the term extracellular vesicles, as you all know, refers to particles that are released from cells, delimited by a lipid bilayer and cannot replicate on their own. That is, they do not contain a functional nucleus. Extracellular particles include non-vesicular extracellular particles and vesicular ones, including extracellular vesicles, synthetic vesicles, and artificially derived, artificial cell-derived vesicles. Without the claim of completeness, let me just show you a few examples for non-vesicular extracellular particles, MVEPs. By definition, according to MICEF 2023, they are multimolecular assemblies that are released from cells, and they do not have a lipid bilayer. They include lipoprotein particles surrounded by a single phospholipid layer, nucleosomes, ribonucleoproteins, exomeres, supermeres, release granulocyte granules, virus particles, protein aggregates, protein complexes such as immune complexes, antigen-antibody complexes, all kinds of crystals, released extracellular nets released by the eosinophil granulocytes, which are larger and more dense, and smaller neutrophil nets, calciprotein complexes, microplastics, sadly, these days is an extracellular particle we have to consider in the investigated tissues. The artificial cell-derived vesicles, again, according to the MICEF 2023 guidelines, are produced by induced cell disruption, such as cell extrusion. So you expose your cells to filtration through given pore size filters, and at the end of the day, you end up with a preparation of membrane-enclosed structures, which are artificial cell-derived vesicles. Synthetic vesicles, according to MICEF 2023, are synthesized de novo from molecular components or made as hybrid entities, such as EV-inspired vesicles or hybrid EVs. When we talk about extracellular vesicle heterogeneity, first we have to consider the tremendous heterogeneity of the releasing cells. As you know, we have approximately 200 different types of cells in the human body. And they all release different kinds of vesicles, but we also have to mention that these cells can each be either in steady state condition, can be activated, dying by different cell death mechanisms, can be tumorously transformed, they can be migrating or senescent. And then the diversity is further increased by the heterogeneity of biogenesis routes, which ultimately lead to a tremendous biophysical and biochemical heterogeneity of EVs. And then I still didn't mention the combinatorial diversity, which is due to the fact that in the tissues, extracellular vesicles come in combination with one another, in combination with different soluble mediators such as growth factors, cytokines, and hormones, and the non-vesicular extracellular particles. Finally, I would like to add that an additional level of complexity is given to EVs because of the EV corona, the biomolecule corona, that is built around extracellular vesicles that are secreted into the extracellular space. As soon as they are released, they become spontaneously surrounded by biomolecular corona that contains proteins, nucleic acids, lipids, even lipoproteins. And I have to also add that some of the corona components might be already there inside the cell. So the released vesicles might already contain some membrane-attached, membrane-adsorbed corona components. When the question comes, which organisms release EVs, the answer is quite simple. According to our knowledge, every single tested organism has been shown to release extracellular vesicles. Of course, the best studied organisms are mammals. Most studies have been conducted in humans and mice, and some data are already from primates. But we also have fish, and I have to emphasize that most data we receive from zebrafish studies. Birds have been shown to have extracellular vesicles. EVs have been shown in avian fluids like egg yolk and serum. Fungi are known to release vesicles. Insects, especially Drosophila, have been investigated, and it has been demonstrated that we find extracellular vesicles in insects. In the non-parasitic roundworm C. elegans, we have extracellular vesicles. Parasites, Leishmania, Trypanosoma EVs are known to modulate host immune responses. Plants are heavily tested these days, and we know that EVs are released to the apoplast and the rhizosphere, and there is defense signaling associated with these extracellular vesicles, and they contain RNA which can silence genes in pathogens. We have to add a very important category, bacteria, bacterial EVs. Those released by gram-negative and gram-positive bacteria are intensely investigated, and we know that they play important role in biofilm formation, pathogenesis, and horizontal gene transfer, just like in the case of archaea, where we also have involvement of EVs in the horizontal gene transfer, also in antiviral defense, stress response, and detoxification. Extracellular vesicles can be classified to the category of ectosomes and exosomes. By MySEF 2023, ectosome is a biogenesis-related term indicating origin from the plasma membrane. All these structures might fall into this category. And exosomes are vesicles which originate from the endosomal system. Exosomes have been classically described as those generated by the exocytosis of the multivesicular bodies, but by now we have the recognition that there are many different pathways leading to exosome production, and I'm sure in the next talk, in the Biogenesis talk, you will hear quite a few details about this. The arms, arrestin domain containing protein 1 mediated microvesicles, are the ones that are originating from the plasma membrane. Thus, they are small ectosomes with a diameter roughly 45 nanometer. We cannot immunostain them for CD63. They lack CD63, but we do have the ability to use an anti-ARRDC1 antibody to immunostain them. They also lack LAMP1. Notch receptor can be recruited to these arms, and they are considered as promising delivery platforms. Another category of small ectosomes has been recently described by the group of Clotilde Thery. In this case, the tested HeLa cells were shown to release small ectosomes which carry CD9, basogene, and the solute carrier protein 3A2, while the exosomes are considered to be the ones which carry CD63 and LAMP1. So these vesicles are definitely ectosomes, even though they come in the size range of the previously described exosomes. Microvesicles are released from the plasma membrane. They are ectosomes, which are larger in size, approximately 200 to 800 nanometer in diameter. They are definitely produced in lower number than small EVs. They are best characterized in the blood plasma, and historically they have been termed microparticles, and you can palette them at 10,000 to 20,000 G. Apoptotic bodies and beaded apoptopodia. These are the apoptotic bodies, and the other side you see the beaded apoptopodia have been extensively described by the group of Ivan Poon. Apoptotic bodies are large ectosome in the size range of 1 to 5 micrometer. They are characterized by phosphatidylserine exposure in the outer membrane leaflet. They may carry nuclear proteins and DNA, and they can be enriched in carreticulin, and their membrane can disintegrate, that is, they can undergo secondary lysis. Large oncosomes have been first described by Dolores Divisio in 2009, as 1 to 10 micrometer sized ectosomes, which are released from non-apoptotic invasive tumor cells. It has been shown that they carry MMPs, RNA, cavalin 1, and ARF6, and MIR1227 has been shown to promote their shedding. Exopheres are large ectosomes in the size range of approximately 4.2, 4.5 micrometer in size. They have been first described by the group of Monica Driscoll, and they harbor protein aggregates and organelles, both which can be degraded or some are non-degraded. And it has been shown in C. elegans that proteotoxically stressed neurons which release these exopheres, they function subsequently better than those which do not release the exopheres. Microsomes are, without any question, a very attractive target for research. Liu's laboratory described them as 0.5 to 3 micrometer ectosomes, which are typically located at the intersection or the tip of retraction fibers that migrating cells leave behind as they move along the matrix. Their marker protein was reported to be tetraspanin 4. ROC1 is a regulator of microsome formation, and they carry spatial information since they might mark the track of the migrating cells. I would like to introduce two flow-induced protrusions in the circulation. One is the FLIP-R category described by the group of Pia Celia-Andel and Rink Nuveland's group. FLIP-Rs are elongated structures which can be as long as 250 micrometers, and they are derived from platelets. Their name says platelet-derived flow-induced protrusions. And phagocytic cells, such as monocytes or polymorphonuclear neutrophil granulocytes, roll over these FLIP-Rs, and they actually take samples from them as microvesicles, and they become activated upon uptake of fragments of the FLIP-Rs. Neutrophil ends are shear-induced, again, elongated processes. Neutrophils, as they roll along the endothelial cell surface, they pull behind them very long cytoplasmic and membrane enclosed structures. These are the ends which ultimately will fragment and round up to form vesicles. They are negative for the canonical tetraspanins, and in sepsis, their concentration is shown to be 100-fold higher than in control individuals. An interesting type of extracellular vesicles has been recently described by Julia Patterson and her co-workers. On the surface of endothelial cells, she observed some very interesting structures that she termed multi-compartmented microvesicles, MCEVs, which contain multivesicular bodies, and she could show that these internal multivesicular bodies from the ectosomes could even exocytose and release exosomes. Mito-EVs are vesicles that carry either damaged, red-colored, or intact mitochondria. And as you can see, there are many different vesicles that fall into this category. Migrosomes, even those can carry some damaged mitochondria or mitopheres or mitolysosomes released by exocytosis, or selective autophagy might release vesicles with damaged mitochondria. Certain microvesicles can encapsulate intact and partially damaged mitochondria, and even exopheres have been shown to contain, again, either intact or damaged mitochondria. Recently, an interesting category of EVs has been described. When we test cells and cell-derived vesicles in tissue culture, we collect vesicles from the conditioned medium. However, in this study, vesicles were not only separated from the liquid phase, from the conditioned medium, but also from the underlying extracellular matrix. And surprisingly, what the authors found was that the liquid phase EVs and the matrix-bound vesicles differed substantially in their lipidomic composition and also in their RNA content, suggesting that these matrix-bound vesicles indeed reflect a distinct and unique subpopulation. I would like to add that we have extracellular vesicles which represent nucleus-free large EVs. One example is the polymorphonuclear cytoplast. As you know, neutrophils undergo netosis. They expel their DNA content. And there is a type of netosis which is a non-suicidal netosis, after which these huge cytoplasts remain on the spot, and these nucleus-free cytoplasts remain metabolically active. They have been shown to be able to phagocytose these bacteria. And to some extent, they resemble blabysomes, the recently described large EVs, which contain different organelles and which have been shown to both secrete and take up vesicles. And it has been shown that these blabysomes are continuously produced not only by cancer cells, but also by normal human and mouse embryonic fibroblasts. I listed a lot of different types of vesicles. Can we somehow group them, find some order and some shared features among them? I would like to highlight just a few of those. Of course, we mentioned that there are some endosome-derived extracellular vesicles. Those are small EVs. We also touched upon the budding or blabbing-derived ectosomes, shedding from the plasma membrane of cells. We have some protrusion-derived EVs, where the fragmentation of these long processes lead to the generation of vesicles. I would like to highlight that we now know a lot of autophagy-related vesicles. We have these vesicles, the blebisome cytoplasts, which are anucleated cell-like structures, and this raises the question, could we consider that platelets and perhaps red blood cells are also very special large EVs? And also we have some pomegranate-like EVs. And in this pomegranate-like EV category, I would like to point to the spherosomes, which are observed in gastrointestinal tumors and glioblastoma, the secreted amphisomes, and the migrasomes, which all share the pomegranate-like structure. And it's impossible not to notice the resemblance to the membrane-clothed virus clusters, which are released either in microvesicles or during secretory autophagy, or even during exosome secretion. Finally, I would like to mention that the Thornback mechanism of small EV release, when a large membrane-surrounded bag is released by ectocytosis, and secondarily its membrane undergoes rupture and the inner vesicles escape, is not only characteristic for the secreted amphisome release, but we also observe similar thing with the spherosomes, and even for migrasomes it has been described as a mechanism of the release of the inner vesicles. The last question, can EV types provide clues about the releasing cells? And of course, yes, very special molecular signatures point to the bacterial or fungal or plant-derived vesicles, their bacterial, fungal, or plant origin. But also, when we look at the mammalian cells, we can find out that when we have steady-state cells, they are characterized by secreted amphisomes. When the cells are activated, exosomes and microvesicles are typically released. Dying cells are known to release apoptotic bodies, necroptotic bodies, pyroptotic bodies. Tumoral cells are the ones that release large oncosomes. Migrating cells release migrosomes, and senescent cells release the senescence-associated secretory phenotype-related exosomes. And with that, I would like to thank you for your attention, and I take any questions. Thank you.
Thank you, Edith, for this nice introduction to the EV world. We have time for a few questions. If you want to ask a question, just go to the microphone. Right here in the middle. And on the left or right. Maybe I can start, Edith, about all this spherosome and, all this torn bag. So what is exactly the nature of the vesicles that is inside the bag?
What we do know that we have some vesicles which have CD63. Therefore, we assume that they are of multivesicular body origin, and they can be classified as regular exosomes. And also, what we do know for the ectosomes, the secreted amphisomes, that also some particles or vesicles carry LC3. I'm not sure if we know all the molecular markers for migrosomes or spherosomes.
Okay. Thank you. We have one question. All right.
Hello. Yeah. Edith, over here, towards your right side. So over here. It's okay. No matter. Great talk. A lot of information. I have a quick question regarding nomenclature of EVs. So one thing that always confuses me as someone who is very new to the field is like how you name an EV. Like we have like ectosomes. We used to call large EVs and small EVs. Now going through your talk, there have been like multiple kinds of EVs and there are different markers. So if you find a new nanoparticle which has been shed by a cell, how do you name it? Do you go by markers or do you go by their biology?
Can you, there is an echo in the room, so can you repeat the last sentence, please? So how do I know that?
How do you name a nanoparticle? Is it based on the biology or is it based on the markers which actually stuck to the membrane?
Well, I think it's something that we definitely have to do a molecular characterization of the newly identified particles. And then if it turns out that the molecular markers and the biological effect is the same as something that has been described earlier, then we now know that we are finding the same particle as before.
I think I have like more follow-up questions. I can come to you later for those actually. Thank you.
Thank you.
Thank you, Edith, again for this great talk. And we move on, and it's our great pleasure to welcome Clotilde Thery from Institut Curie, who will tell everything about biogenesis and secretion.
Yeah, thank you for inviting me to give this presentation on biogenesis and secretion. Well, I must say that I mean, what I'm going to present is the way I see the field and its evolution. Maybe some other people will not see it the same way, but this is science, right? So some of my slides will be redundant with those of Edith, but of course, I mean, it's always good to have several times the same information so that it sticks better to your mind. So this is just to reintroduce the idea that EVs are very heterogeneous, that some of them come from the plasma membrane, and they are called ectosomes. Some others come from multivesicular bodies and are secreted when these multivesicular bodies fuse with the plasma membrane to release their internal vesicles outside, which are then called exosomes. Viruses, enveloped viruses, should be considered as extracellular vesicles if they have a lipid bilayer surrounding their internal capsid, and they can form either at the plasma membrane or in multivesicular bodies, depending on the viruses. So this is again to re-stress that the term small EVs, and you have to define yourself what's the size that you use to say that an EV is small, but generally it's around 150 nanometer, the limit, and this covers both exosomes, which are small because that's the size of the intraluminal vesicles of MVBs, but also the smallest ectosomes coming from the plasma membrane. Whereas large EVs are the ones that are larger than that, and these large EVs, they are mainly coming from the plasma membrane, where there is no size limit in their production. And of course, always remember when you are analyzing extracellular vesicles that cells do not secrete only extracellular vesicles, but lots of other things that you may recover in your preparation of vesicles, such as non-vesicular particle, exomeres or others, and also, of course, soluble factors. So this is an image from an eukaryotic cell releasing EV, but of course eukaryotic cells are not the only type of organism, and we have also, of course, EVs released by non-eukaryotic cells like bacteria and archaea. And what this slide is just to remind you that since bacteria and archaea do not have intracellular compartments, they cannot produce exosomes because exosomes are formed in intracellular compartments. The bacteria EV, they can come from either the outer membrane with or not some of the inner membrane. This is for the gram-negative bacteria. Or they from gram-positive, which only have one membrane covered by a peptidoglycan wall. The EVs, they form from this membrane, and they have to somehow cross the wall. And another way of EV to be released by bacteria is from sort of explosion, a bit like apoptotic or necrotic bodies of eukaryotic cells. And I just wanted to put in this slide something that came to my understanding very recently, because I was not even aware that we had another kingdom than bacteria and eukaryotes, which is the archaea. So the archaea are these other non-eukaryotic cells that are actually closer to eukaryotes than bacteria. And indeed, these archaea, they also have been shown recently to secrete EVs that come from their membrane, because again, they don't have internal compartments. But this secretion apparently seems to be also due to molecules or molecular machineries that resemble those used by the eukaryotic cells, and I'm going to come back to that in the rest of my talk, but such as escort protein or small GTPases. So it's a very interesting intermediate organism between bacteria and eukaryote. So the rest of my talk will be about eukaryote, and I have not split the eukaryote into all these different types that Edith very nicely presented before. So this is a generic image of an eukaryotic cell. So as Edith said, as I said, eukaryotic cells secrete EVs from many different parts of their structure. And many different groups like focus on one or the other, like for instance groups like those of Guillaume Van Niel, Michael Pertel, Pascal Zimmerman, Alyssa Weaver, Derek Gibbons. They focus mainly on the exosomes, the one that form from internal compartments containing intraluminal vesicles. But there are also groups focusing more on the extension-derived vesicles like cilia, nanotubes, or philobodia. There are several groups that are particularly interested in the large vesicles that come from the plasma membrane. Again, all these names have been extensively described by Edith. And the most recent one is the blebisome, indeed, that was published this year by Jeppesen in Cell, which are like anuclear cell-like structures. And more recently, I mean, in the last, I would say, 5 years, we had really the knowledge that a lot of vesicles can come from an autophagy derived mechanism, an autophagy dependent secretion. So that was illustrated in this review by Guillaume Van Niel not so long ago. And I think the first mention of this was from the group of Debnat in 2020. And more recently, the group of Edith Buzas, and she described it also, showed this amphi-ectosome release of fused compartment between amphysome and multivesicular bodies that lead to encapsulated MVBs, which then release their internal content. So these are really so many types of vesicles that you've heard about in the previous talk. A very big complexity is that once these vesicles are outside the cell, they are really difficult to separate from each other because they really have overlapping physical properties. The size of these extracellular vesicles, which is illustrated here on top of this slide, overlaps between the size described for exosomes, between 50 and about 150 nanometers, which is the size of intraluminal vesicles of multivesicular bodies, but it really overlaps with the size of the smaller ectosomes, also of the smaller apoptotic bodies. It overlaps with the size of retroviruses and with the size of some lipoproteins, while other lipoproteins may be smaller. And at the bottom of this slide is the density that's illustrated, because many protocols that you're going to hear about in the second part of this talk, they use density gradient to separate the subtypes of vesicles, but here again with the same color code as on the top part, the density described for these different vesicles also do overlap in many ways. So you cannot really define one vesicle once you isolate it from a biofluid. You cannot use its size or its density to say that it was an exosome or it was an ectosome. So really the only way to really know if a vesicle was coming from the inside of the cell or from another part of the cell is to follow a vesicle with live tracking. And this has been done actually a lot with different molecules, like molecules that had been shown to be specifically enriched on different types of extracellular vesicles, like for the migrasomes, the T-span4 was shown to be enriched, a marker of this migrosome. For cilia-derived EVs, the molecule PKD2 was also used as a marker, or hyaluronan synthase 3 for the long plasma membrane-derived protrusion. So the idea for these approaches is to use these molecules and tag them with a fluorescent tag, and then you can follow the way these molecules go away from the cell, and this way you can see if they come from the plasma membrane, or if you use markers that are more enriched on like multivesicular body-derived intraluminal vesicles, you can also see that. And the groups of both Michael Pertel and Alyssa Weaver have used the fluorescence properties of GFP, which is quenched by acidic pH, so that there is no fluorescence on this molecule when the molecule is inside in late endosomes, and the fluorescence appears when this late endosome fuses with the plasma membrane, so that they have used that fusing this pH-sensitive green fluorescent protein to CD63 to really visualize and quantify the exosome release. And there are many other methods and described in this nice review that was published a few years ago, so you can have a look at this for more ideas. But of course, I mean, these are not possible to do in a very large scale for large-scale production. We, in my group, we have used also live tracking to try to distinguish the fate or the biogenesis of EVs expressing either CD63 or CD9. And so this study we started because at the time, at the time, both tetraspanin CD63 and CD9 were routinely used as marker of exosomes, but we always thought it was a weird idea because when you look at a cell, and this here is an HeLa cell, when you look at the distribution of CD9, it's really clearly at the plasma membrane and very little in internal compartments, whereas CD63 is mainly in internal compartments, which are by electron microscopy, you can see there are multivesicular bodies. So how could these two different locations lead to identical exosomes? That was the question. So to try to answer this question, we used this rush approach to follow the trafficking of either CD9 or CD63 by controlling it, by stopping it in the endoplasmic reticulum through this rush approach. Again, I'm not going to describe it. You can look at the paper. And when you release the EV, the molecule from the ER, you follow their trafficking and you see where they go and if they go to the plasma membrane or to MVB. So that's what we did. We also used a mutant of CD63, which had been described 20 years before as with the tyrosine signal in the C-terminal domain, which is the one that targets CD63 to MVB. If you mutate it to an alanine, then CD63 is not anymore in the internal compartments here, but much more diffuse and distributed in very small intracellular compartments near the plasma membrane. And when we looked at the EV release from these cells expressing either wild type or mutant CD63, we paletted EVs by centrifugation, differential centrifugation, and we could see that with the wild type CD63, we did not recover much CD63 in the pellet. We recovered much more when we were using cells expressing the mutant CD63 that's more at the plasma membrane. So that really suggested that in these cells, the HeLa cells, there were much more EVs released from the plasma membrane than from the MVBs, so much more ectosomes than exosomes. So, and we also had this observation based on the rush live intracellular trafficking. So this was just to raise the idea to you that exosomes are not, the MVB, multivesicular body-derived vesicles, are not necessarily the most abundant vesicles that are secreted, and depending on the cells, it may be rather the ectosomes that are more abundantly secreted, and it really depends on the cells. So now, how can you try to identify the subcellular origin of EVs? A lot that has been done by many groups, and it's actually a question I get very often when I do a presentation, is how can you block secretion of exosomes, for instance? So generally what people do is to try to use things that will inhibit some intracellular machineries, and these molecules can be drugs, can be small interfering RNA or small hairpin RNA or guide RNAs to inhibit molecules that are known to be involved in intracellular machineries and trafficking, such as those that are highlighted here, the escort proteins, which are involved in the formation of multivesicular body intraluminal vesicles, but also in budding of vesicles from the plasma membrane. There are also the sphingomyelinases that are involved in formation of vesicles, both at the plasma membrane and in MVBs, and other proteins. So if you inhibit any of these proteins with drug, siRNA, shRNA, then you can inhibit release of some of these vesicles. But what's really important to question is whether you also affect the release of some other vesicles or some other particles that we were not really following in your mind. So it's really important to ask this question, and I think that I saw very recently a very good example of that. For instance, the drug JW4869, which is an inhibitor of the neutral sphingomyelinase that blocks formation of ceramide and budding. It was described in 2008 to block release formation of multivesicular body intraluminal vesicles and release of exosomes. But it was shown later that this drug, while blocking this exosome or small vesicle release, was also increasing the release of large extracellular vesicles. And now, very recently, was also shown to reduce the release of small ectosomes derived from Philopodia. So it really shows that JW4869 is not an exosome-specific inhibitor, as it is still often used in the literature. So please keep that in mind in your own studies. And then for other types of machineries, like there are all the small GTPases, the RAB proteins that are involved in trafficking between internal compartments at different levels. So a long time ago, back in 2010 actually, we had shown that RAB27A and B were really crucial to allow secretion of exosomes by HeLa cells. And more recently, we also showed a few years later that it was not only required if you inhibit RAB27A or B, you were also inhibiting secretion of some soluble proteins. So that would change also the physiology of your cells and the features of the secretome of the cells. And also, it was surprisingly shown by actually Matthias Ostrowski group, who was the first author of the paper and then generated his group in Argentina. He unexpectedly observed that when inhibiting RAB27A, he was inhibiting exosome release, okay, but he was also inhibiting formation of formation of lipid-enriched structure at the plasma membrane, and thus inhibiting release of budding viruses from the plasma membrane. So this was not only inhibiting exosome release, but also release of ectosomes, these viruses. So again, no specificity of RAB27, no real specificity exclusive to exosome release. Now there are other RAB proteins like the RAB11 and RAB35. They have been shown to affect the release of vesicles from earlier endosomes, earlier than multivesicular bodies. And affecting the fusion or the recycling and the secretion of vesicles from these other compartments. The small GTPases RAL-A and B, they are also regulating biogenesis of MVBs and exosome secretion. But really what's not so clear to me is what other function, what other secretion of the cells these molecules regulate. A recent development on exosome biogenesis is this interesting observation by the group of Guillaume Van Niel, but also by the group of Alicia Weaver, that there is a need for fusion, membrane fusion contact sites between endoplasmic reticulum and multivesicular bodies to change the membrane of these MVBs and allow them to become competent for fusion with the plasma membrane, or to allow targeting into the intraluminal vesicles of MVBs of specific components such as RNA and RNA binding molecules. So, well, please go and have a look at this paper if you're interested in these types of biogenesis. So I just want to finish with a few summaries and recommendations. So really what makes the field complex, and it's not easy to answer easily if a vesicle is really an exosome or something, is that there is really no means to specifically inhibit or promote the release of a given EV subtype without affecting many other intracellular trafficking pathways or many other properties of the cells to secrete molecules. So keep that in mind. If you use one of these molecules to inhibit exosome or ectosome or EV release, you will most likely inhibit or promote many other secretions. Another difficulty is that there is no generally accepted specific markers of a given EV subtype. Of course, some molecules are enriched in multivesicular bodies like CD63 and LAMP in the cells that secrete them. And so they are relatively good to be used as proxies of markers of exosome, but still keep in mind that these molecules, they go at some point through the plasma membrane, and they can thus end up being also present on ectosomes. Then if you talk about vesicles from internal compartments, there are also not only multivesicular bodies, but also earlier endosomes that are a source of ectosomes. There are also the autophagic compartments. And again, I mean, for the ectosomes, there are some molecules that are very enriched at the plasma membrane, like CD9 or others that may be proxies of ectosomes, but also these ones that can also at some point traffic through the endosomes. And another question is the non-vesicular extracellular particles. Do we know anything about their biogenesis, where they are from? Are they formed in the secretory pathway, like the ER, Golgi, or secretory vesicles? Are they coming from broken cells or large ectosomes? Or are they coming from the culture medium? This is still open questions. I think we have not really advanced so much on this type of questions. As a recommendation, when you want to manipulate your cells to affect secretion of a given subtype of EV, don't consider that results that have been published by other groups on other cell types apply to your cells without verifying it yourself. All cells react differently. Some will undergo apoptosis when you treat them with the treatment, some will not. And so it's really necessary to first do this treatment on your cells and see how they react, not only for your vesicle of interest, but also for the other. So you need to really analyze the consequences of manipulating the secreting cells on other physiological function of these cells, and analyze the composition of all the EP subtypes that you can think of, analyze the viability and the senescence of the cells, and analyze an array of secreted products, not only EVs, but also the secreted molecules. And well, for what we should develop, what we should keep working on, is to know more on the biogenesis and in particular of the non-vesicular extracellular particles. So for some, like the lipoprotein, it's probably been studied a lot by people working on lipoproteins, but for the others that are more recently described, it's not so clear. To know more about the respective contributions of extracellular vesicles and non-vesicular particles, and also soluble factors, including those that make the corona that Edith described, in the physiology and pathology of EVs as now described in most papers. And now to define EV subtypes, since it's really difficult to know the biogenesis and the biogenesis pathway, and that maybe to answer the question that was asked to Edith, maybe it's easier to define EV subtype by a surface protein that they express, if you are interested in a particular surface protein, rather than really defining by their biogenesis pathway. At least it's safer. This you can really analyze and show. Okay, and I'm going to stop here. Thank you for your attention, and if you have any questions.
Thank you, Clotilde, for this excellent overview. We have time for a few questions.
I can start with one, Clotilde. So what about polarized cells, and especially in their own environment, like neurons or epithelial cells? Is there even more complexity into the biogenesis and secretion?
Well, yes, of course there is. I mean, polarized cells, yeah, you're right. I could have put that in my presentation. I didn't think of it, okay, for next year. But indeed, there have been studies like for epithelial cells that are polarized in apical versus basal condition that do release different EVs at the different places. For neurons, it's probably even more even the case also. And the machineries of secretion are different from the two sides. So indeed, another complexity that I didn't even allude to.
There's one question in the middle.
Yes, thank you. Really intriguing talk. Adding to the complexity of the different subtypes of the EVs coming from different sources or origins, so I was wondering that is there any data or research on the secretion of the EVs and how they kind of form on the plasma membrane? So in theory, you could have membranes budding like so that the inside of the vesicle kind of turns around and you would have two types of vesicles. So I'm thinking of the surface protein thing. So if you have the surface protein actually inside of the vesicle. So I was wondering, can you comment on the budding of the EVs from the plasma membrane?
Well, the budding of the EVs from the plasma membrane, theoretically, I mean, in textbook knowledge, it should be exactly similar orientation. I mean, the extracellular domain of the transmembrane protein should remain outside. And it's actually the same for the multivesicular body because the budding orientation is the same as from the plasma membrane. So there have been lots of studies also on the budding from the plasma membrane. I cited a few groups in this presentation. Yeah. But you're right that there are some observations that there can be some inside-out membrane molecules whose extracellular domain seems to be inside EVs. It's been shown by a work of the group of Jan Lodval already, I think, 10 years ago. Yeah. It seems to be really a minority of the proteins, so probably a minority of the vesicles. I'm not sure how this can happen, so that's also something that would need studies to understand the biogenesis and the inverted membrane orientation of some vesicles. So to me, it's still a completely open question.
Yeah, thank you.
Thank you, Clotilde. So we know about different types of EVs, how they are generated and released from the cell. Now it's also time to find out where they go. So it's our pleasure to welcome Dr. Charles Lay to introduce us to this.
Thank you very much. Sorry, let me find my pointer again. So thank you, everybody. I would like to thank the organizing committee for inviting me to give this education talk. And to be very honest, when I first received the invitation to go talk on biodistribution, I was like, sure, I've been working on this for about 15 years. I can do it. Then about a couple weeks ago, when I look at the program, I was like, are Edith and Clotilde before my talk? Did you actually put me here? After two of the most admired scientists, which I admire. So I'm very, very honored to give a talk after them. So let's go on. So I used to love watching movies before I become a PI. And when I was looking at vesicles, it's like Life of Pi, that you try to track its journey. And I believe that Clotilde, as well as Edith, gave a very, very comprehensive overview of vesicle biogenesis, as well as the different cell populations. But what happened after they have been born, or they have been generated? So supposedly, the vesicles would be distributed, be it locally or distally, to different parts of the body. But if you're looking into vesicle as a therapeutic product, then you'd be interested as to where they go about. So that would go into biodistribution and clearance. Now, when they have received by the organs or tissues, then they go into the uptake. And finally, you would be very interested about the fate of the vesicle itself, as well as its cargo. And finally, I think that's why people are interested in vesicles, is that they could elicit biological effect. And when I first entered this field, I think this is where we start. We usually saw an uptake, and we saw a biological effect, and we got super excited about it. And it was only until later that people became more interested about the biodistribution and clearance, and also the cargo delivery efficiency of the vesicles. So moving forward, I will start with the biodistribution and the clearance part of the vesicles. Now, I don't consider myself a very smart guy, so I usually try to simplify the talk. And because every time when I am in an education session, I try to consider myself as someone who just stepped into this field. And so for the purpose of this talk, please forgive me if you think it's really simplified, because I try to simplify it. I try not to mention any of our personal work, and I actually don't mention many studies, but instead I try to kind of provide an overview or a guideline as to what worked for us and what did not work for us. So what are, or why do you want to do vesicle biodistribution? Is that most of the studies you might have witnessed earlier is that they focus on the origin, the composition, and production of vesicles. And if not, they would be very interested about how those vesicles affect the body. So essentially, that would be the pharmacodynamics of the vesicles, and the effect may be desirable or undesirable. Relatively speaking, I see less study on pharmacokinetics. So essentially, is what your body does to the vesicle or body, the drug. And biodistribution is part of pharmacokinetic study as well as the uptake. And they are quite important to understand the mechanism of action of vesicles, be it for therapeutic purposes or for development. And also, vesicles may be toxic. I think a lot of people didn't want to look at it that way, but too much of everything may not be a good thing. Now, again, vesicle biodistribution is important in our understanding of basic biology, such as development. I should put a plant here as well. I know that vesicles is really hard to be studied in vivo. And I think to understand how they are distributed dynamically is really critical for our understanding as to how vesicles contribute to development as well as disease progression. They are also very critical for clinical translation, especially in the preclinical part of the clinical translation process. So what is vesicle biodistribution? So in essence is quantification of vesicles as well as its distribution in vivo, such as the capybara that a lot of people might like. And believe it or not, I just learned this from last week going to the zoo that they are the largest rodent on Earth. And people have actually tried to use them as an animal model in their studies. And that will involve tracking vesicle in terms of space, time, and for resolution, it depends on the question you're trying to ask. It could be at tissue level resolution, or it could go into cellular level, or further, some people might be interested to the subcellular location of the vesicles once they have been taken up by the tissues. And I try to categorize them by two general categories. One is exogenously administered vesicles. And there can be many different routes of administration, including the most common one is IV, intravenous, or intraperitoneal, subcutaneous, or topical. There are many ways to administer them, and another consideration to take in mind is how much vesicle you try to administer and track them. Another category I will put is endogenous vesicle. I think the most common way people try to look at is if they can engineer tissue to release the vesicle and specific tissue driven by specific tissue promoter, and also to track them over time. And these are actually quite difficult, especially for the endogenous part of the vesicle release. And how do you track vesicle distribution? I think the first or the most obvious way is to image them, and another way is to dissect them, or you can actually do quantitative PCR to track them if you can somehow identify a signature within the vesicle, which is not very easy. I think importantly, we now realize that it's best if you can use multiple methods to validate your results. And I would say that orthogonal analysis is really important when it comes to biodistribution study. So try not to just use one way or one technique to measure vesicle, especially for biodistribution. And this is just some tracking strategy that I would recommend when you want to study vesicles biodistribution. Number one is to identify your vesicle question. This could be ranging from development to disease, devices, biomarkers, as well as therapy. You'd be surprised, a lot of people are just really fancy by the images without thinking about what question they're really trying to answer, and I think this is really key. Then the second part of the planning process is to identify your in vivo model. So you have learned that vesicles come in very many different sizes, and they come from many biogenesis pathways. Then how do you want to apply and visualize them in the different in vivo model? Because they come in different sizes. The C. elegans is about 1 micron. In non-human primate, they can be up to 55 centimeter. So this will ultimately determine the resolution that you need. So after the first and second part, you need to think about the specificity of your vesicle labeling or label-free. There are, I think, from Stephen Bopar's group that they can actually do second or third harmonic generation to visualize vesicle, at least ex vivo. You also want to consider about the accuracy that you require in terms of space and time to measure the vesicles in vivo, and further the resolution that's required, whether you want to visualize the vesicles in the whole animal resolution or at subcellular resolution. And importantly, you also want to consider about providing sufficient signal-to-noise ratio when you're trying to track the vesicles in vivo. And I will refer you to session 3 after this session. And I think Dr. Guillaume Venille has also very nice talk on this that he will discuss about the different labeling strategies that have been developed to date. Now, due to time, I also only go over a couple really nice study, in my opinion, in exogenous vesicles in large animals. And for endogenous vesicles, I will go over one example where they track vesicle release in zebrafish. So this is a very elegant work done by Verweij in zebrafish, where they use CTC3 pH lowering. It was initially developed by Elisa Weaver's lab, and I think Clotilde introduced this reporter already. And in addition to that, they ubiquitously expressed this reporter in zebrafish and found that there is an inter-organ EV communication in the zebrafish, where yolk syncytial layer release vesicles actually go into the blood circulation and gets captured by macrophages and endothelial cells in the caudal vein plexus in a dynamine and scavenger receptor dependent manner. And for exogenous vesicle biodistribution in large animals, this is in collaboration with Dr. Ken Whitworth's group by Tom Drindox, and I think he's also one of the speakers in the later session today, that they apply our reporter, which is a bioluminescent and fluorescent-based reporter. We coined the term PONGRET. And then by collecting vesicles released by XPI 293 fluorescent vesicles, they were able to track the distribution of administered vesicle through intravenous or intranasal in the mouse as well as a macaque model. And further, they actually tried different dosages and did a pharmacokinetic study to elucidate the biodistribution and clearance following the administrations, and further found that the vesicles can be taken up by peripheral blood mononuclear cells. And in this case, they found that B cells took up a lot of the IV administered vesicles. So we cover most of the biodistribution and clearance. Now I'll try to go into EV uptake. In terms of the mechanism of EV uptake, it can be generally categorized into several categories. One is the ligand receptor interaction. So if you have ligand on the surface of the vesicles and interact with the recipient cell's receptor, then there would be initial uptake or docking. In less occurrence, you might see membrane fusion. In this case, the vesicle will fuse with the membrane, and it will release its cargo into the cytoplasm in the recipient cells. They don't get observed as often. Instead, most of the study, and this is a very nice review by Collison, it was published in JEB in 2024, that most of the vesicles are taken up by all these endocytosis pathway, ranging from clathrin-mediated, caveolin-mediated, or clathrin and caveolin independent pathways. CLIC is clathrin independent carrier, and this is a long term. GEEK is GPI anchor protein enriched endosomal compartment. And you can also see phagocytosis and micropinocytosis. So these are all the pathways that people have reported vesicles to be taken up by. But again, it's really complicated, and I don't think I'll have time to go over all of these. But I want to share with you that they did almost like a meta-analysis that from 26 studies encompassing 7 cell classes with 21 normal cells and cancer cell lines, what they have found that there are 3 most commonly observed vesicle endocytic pathways, and they are caveolin dependent. So in this case, 14 out of the 21 cell lines took up the vesicles through this pathway, and 12 of 21 took up vesicles by clathrin dependent, and 11 out of 21 cell lines took up vesicles by micropinocytosis. On the other hand, lipid wrap dependent, which are caveolin and clathrin independent pathways, only stand about 2 of 21, whereas only 1 of 21 was found to have membrane fusion and for phagocytosis as well. So this goes to tell you that cells can employ multiple endocytosis pathways, and for 5 of the 21 cell lines, they can actually employ all 3 pathways. About half of them use 2 pathways, and 6 of 21 cells use 1 pathway. So if you think the biogenesis is complicated enough, the way they take up the vesicles is also very complicated and not very singular. Something to keep in mind is that among these studies, different types of vesicles were applied. So again, you have to think a lot about like what entity you're working with exactly. And most of these studies were done in vitro. Okay, what is it like when it go in vivo? I think that will be a whole different ballpark to go into. And in terms of the specificity of vesicle recognition and uptake, I think it largely depends on many factors. One is where the vesicles came from, because it will attribute to their size, as well as their surface membrane protein, as well as post-translational modifications such as glycosylation, as well as corona that it did mention later in her talk. In terms of the recipient cells, of course, it depends on the ligand, the receptors that express, as well as the state that they're in. And I think something to know as well as the environment that the vesicles, as well as the recipient cells are in, is that what is their inflammatory status during the uptake? What is the pH, hypoxia? So those things all play a role during the uptake process. And it's really hard to mimic what in vivo environment is like by just doing the in vivo study. So I think those are things that we need to keep in mind when we're trying to interpret our data. And finally, for EV fate, I think this is where people are really interested, is that I finally have delivered the vesicle to a certain organ, the tissue. Did I actually offload the message or the cargo to elicit the biological effect? Sometimes it'll work, but most of them actually will actually end up in early endosome and finally endolysosomal degradation. So, but in rare occurrences, there are actually something called transcytosis, where the vesicles got taken up by clathrin, caveolin, and lipid raft-dependent endocytosis, and they actually just travel across the cells to be released on the other side. And this was actually, they were witnessed in endothelial cells in the blood-brain barrier, the central nervous system endothelial cells. I don't recall this being witnessed too much in other systems, but for most of the other pathways, most of them goes to early endosome. Or some of them, such as macropinocytosis and phagosomes, they will actually be directed directly towards the lysosome. But a lot of them actually get, I think, predominantly degraded. Some of them actually do escape, and this is one of the most elegant study I have seen where they actually capture cargo release from endosome. So in this case, they decorated the vesicles with GFP CD63. But this is interesting, the CD63 GFP, they actually tag the GFP in the cytosolic side of the vesicle, whereas the fluorine one actually tagged on the extracellular space. So under this setup, you will see that the vesicles are here. When it's endocytosed, if it is not ruptured or degraded, you will see the signal like this. But if it actually, for example, in the multivesicular body, and it actually fuse with the endosome or lysosome membrane, they can actually expose this content. And when this GFP content exposed, the cell expresses M-cherry tag nanobody, and then you can actually see the red as well as the red, the green and the red signal. And this is what they saw, that some of them are not, the cargo has not been released, so you see the green dots. And for the one that have been released, you can actually see the colocalization between the M-cherry nanobody as well as the green vesicle signal. And they also did a clamp, a correlative light and electron microscopy signal, and they found that in this case, this is EM of region 1 to 3, where they show colocalization between the GFP as well as the release cargo. And they could actually see the fusion of the vesicle at the limiting membrane around the endosome and lysosome parts. And this electron dense region is the lysosome. So moving forward, again, so Randy, I borrow from your format from last year's education day. There are many gaps in our knowledge. I think one of them is the vesicle delivery efficiency. It's really hard to know the efficiency of vesicle cargo loading. On the other hand, it's also really difficult to understand how much has been offloaded. And I think that's what a lot of people are trying to work on. In terms of vesicle biodistribution, one question I always have is that doesn't matter how you label it, if you see the signal going to a certain part, are they still intact vesicle? And I think that's something that remains to be elucidated. I think organotropism is something that a lot of people are looking into. I think earlier days, a lot of people are trying to target vesicle to specific organs, and they only look at that organ, which is not how vesicles work. What we have found is that vesicles are very dynamically distributed. If it goes to one organ, it actually can get distributed to other parts dynamically. So that's something to keep in mind. It's about the organotropism of the vesicles over time. And I think another key question that's really hard to answer is, what is the endogenous release rate of vesicle from the different tissues? I think not many people have the right tool to answer this yet, and to be honest, if you know, please let us know. Because what is the physiological vesicle dosage is also a key question, and this may affect the uptake, the fate, and finally the biological effect that people are trying to study. And finally, I would also like to mention about rigor and reproducibility. I think this is also one of the main sessions in this afternoon, is that try to report how you label your vesicles, or it could also be label-free, but please describe how you did it. How did you image your vesicle, and how do you quantify it? And what are the controls that you have applied to come to the conclusions that you suggest? And are there ways that you validated those? I think this is an echo to Clotilde's statement that don't try to convince reviewers with your data with just one type of experiment, because vesicles can be pretty complicated, so forth. And I think session 6 will cover quite a lot on that. And finally, when I first saw this topic on what are they, where are they from, and where did they go, because I used to watch a lot of movies when I was a kid, this movie came to mind. I don't know if you guys have watched it before. If not, it tells you how old I am. It's like, oh brother, where art thou? So spoiler alert, it's about men trying to find the meaning of life, blah, blah, blah. Okay. Now, I love posters because they usually have some comment on what the story is about. And here it says that they have a plan, but not a clue. And when I saw that, I was like, wow, isn't that what vesicle is like? And growing up in Canada, but I also love animation a lot, so I usually like to watch the Japanese translation. This is a Japanese translation of the poster. So rather than, oh brother, where art thou? I just go, oh brother. They only say the brother. They don't even care about where art thou now. And then what I love about Japanese translation is that they not only translate, they also put their interpretation about the story. And if you look very carefully at the very bottom of the poster, it actually translate or put their comment as that it's a funny and exciting adventure story about treasure and love. And I thought this puts vesicle quite well, because I still remember to this day that when I started vesicle research in Dr. Zendra Breakfield's lab, that I did ultracertification, and then I kind of held that tube in my hand. I was like, oh my God, these are exosomes? So remember those days and don't shake it. So I think to this day, when I'm trying to understand how vesicles move about in the body, I still consider them as a pretty adventurous journey. And finally, I think we have come to this part about the biological effect, which I won't talk about, but rather I think it's about time for us to have some coffee break. So I'll be happy to answer any questions. Thank you.
Thank you, Charles. So we have time for a few questions. And while waiting, I can already ask one. So we have heard about different types of extracellular vesicles. What do we actually know? How do they differ in biodistribution and endocytosis pathway that is used?
What do we know about?
What do we know about it? Yeah.
We know very limited amount, in my opinion, unless you have very specific marker, such, for example, the CD63 pH lowering. But in our work, we literally categorize them by size. Yeah, because there are so many biogenesis pathways. Yeah, so I think by size, we found that they distribute differently. We have not tried looking into specific vesicle. For example, like CD63 or other tetraspanin mark vesicles. Yeah, it's to us it's too narrow. Yeah, and not let alone we did not go into specific tissue. Yeah, there were some work that did that. Yeah, I think one was in communication biology. They were tracking vesicle released by the heart, if I remember correctly. There were another recent one, which was also quite nice, that they were tracking endogenously released vesicles. Yeah. But how long does it take to get it distributed over time? I think those are pretty hard questions to answer, because if the signal is not strong enough, you can't see it. Yeah.
Well, thank you, Charles, for the nice presentation. Just one question, maybe to conclude. So when you play with exogenous EVs, inject them in vivo, how do we know that they do not compete with endogenous EVs?
Sorry, could you repeat the last sentence? How do we know?
What about the competition between exogenous EVs that you inject and endogenous EVs that are just doing their job?
This is a really hard question. I don't think we can answer that right away, because it's really hard to see non-label endogenous vesicles and how they compete with them. Is that your question?
Yeah.
I think it's really hard to answer that.
But that should be important to...
It is important. So supposedly you can label endogenous vesicles with one reporter, and then you can inject with another reporter which you label, right? So maybe you can do that. But what confounds me is always the dosage. Yeah, it always confounds me. Is that what is a physiologically relevant dosage? Because in one of our work published in 2023 in advanced material is that too much cancer vesicle can kill the animals. Yeah, and we thought we were the only one witnessing it until people later came to us and said, yeah, we saw the same thing. Yeah, vesicles can kill. But those were cancer vesicles, so it really depends on where the vesicles came from. Yeah.
Okay, so there's still a lot to understand.
I think in vivo is like a black box to many of us, yeah. And we don't even know the specific uptake pathway used by the different tissue and the cells. So I think we still have quite a few ways to go in the in vivo realm. Yeah. Okay.
So thank you, Charles, and let's thank all the speakers from the morning session. And then it's time for a coffee break, and we start again at 9:50.